Much. Good afternoon, everyone. My name is Gloria Kirabo, and a student from Makerere University. I'm going to be presenting results on the first Continental Genome-Wide Association study of blood urine nitrogen and kidney function, and this was done in a cohort in Uganda. This was part of my master's work. So to give a brief background, the kidneys, we all have kidneys and they're filtering organs in our body. Well, kidneys act as filtering organs because we have processes happening in the body over time and we generate waste products. And these waste products have to be eliminated from the body to enable a good environment for processes to keep happening. So blood urine nitrogen is one of the kidney biomarkers that is used in clinical practice. This is because it is one of the waste products generated from protein breakdown. So proteins are broken down and then they are translated to ammonia, and this ammonia is toxic. So in the liver, it's broken down to urea, and this urea is then taken to the kidneys for filtering. So we expect that a normal functioning kidney should be able to regulate the levels of blood urine nitrogen around 7 to 20 milligrams per deciliter. And usually some of the factors that will lead to high levels of BUN may be a deteriorated kidney, and this could be drugs, or it could be the lifestyle of an individual, or sometimes it's genetics. For example, this study was looking into the genetic factors of kidney function. So to give a brief background about the genetics of blood urine nitrogen, to date in the GWAS catalog, we have about 1,872 associations that have been reported with blood urine nitrogen levels, and these are in a total of 40 studies. However, all these are in populations of Europeans, East Asians, and Latin Americans. And to date, we only have 2 Continental Genome-Wide Association studies that have looked at kidney function, and these have looked at glomerular filtration rate and creatinine. We do not have any study to date that has looked at blood urine nitrogen as a key biomarker for kidney function among continental Africans. So when you look at the map on my right, it shows the prevalence of CKD on the continent. Eventually, what we're interested in kidney function is that when the kidneys fail, then you eventually develop into chronic kidney disease, which later develops into end-stage kidney disease. So these biomarkers are used to assess how well the kidneys are functioning. So by 2018, we had traces of CKD all over the continent. On the contrary, to date, the Kidney Gen-Africa project, which was launched in January this year in the FSHG conference, has studies from around 7 countries on the continent. So we have 54, and we only have participants in 7 countries with whom we are looking at kidney genomics. So it shows a very big gap in actually understanding or having participants that can help us understand the genetics of kidney on the continent. So this particular study was carried from the Ugandan Genome Resource Cohort, and this is a cohort that was done in rural southwestern Uganda. This is in a community called Chamulibwa, and this particular population is part of the general population cohort that was established by the Medical Research Council in 1989. So data is collected from this community over time, and particularly the Ugandan Genome Resource data was collected in cohort 22 that was collected in 2011. So this is when now they went ahead to sequence genome data, and that is what we used for this particular study. The blood urine nitrogen levels were assessed using the blood samples collected for these particular participants. So deeper into the methodology, when you're running a GWAS, you literally need the genotype data, and then you need the phenotype. So our phenotype in question was the blood urine nitrogen levels, and then we had the corresponding genotypes of the participants. So we went ahead to look out for the summary statistics, and here we used GEMA, which is a linear mixed model, mostly because it's more efficient and it accounts for a lot of population structure. And then we went ahead to identify the lead SNPs from our summary statistics. And then we used these lead SNPs to go ahead to define mapping to determine the cause of variance. And then we went ahead to do functional annotation, try to find out the loci and the SNPs we're identifying, which genes are they mostly related with. And then we went ahead to do replication. Replication is when you're trying to validate your results, trying to find out if what has been found in other populations can be replicated in your particular study. So from our results, we did not obtain any SNPs at the conventional GWAS threshold. The conventional GWAS threshold is p-value less than 5 times 10 to the power negative 8. According to Bonferroni correction, this is when you test every SNP about 1 million times. So, however, we did not cut out the study. We considered a suggestive significance of p-value less than 5 times 10 to the power negative 7, and we had 13 SNPs with this p-value. So when you look closely, the image on the right is the Manhattan plot. It shows the results on the x-axis. We have all the variants in the population in the different chromosomes, and then on the y-axis, we have the corresponding p-values. And clearly, when you look at the red line, we did not have any SNP that crossed the conventional threshold. So that's why we suggested a slightly higher threshold. So we went ahead to also look at if we had any substructure or bias in our results. And when you look at the QQ plot, it's an evidence of what was expected versus what we observed in our analysis. So the red line shows what was expected, and the black line is what was observed in our analysis. So you can clearly see that we did not have a lot of bias within our study. So it's what we really expected. So we went ahead to do LD clamping from the SNPs that were suggestively significant. This is trying to identify the SNPs that have the lowest p-value in the given loci, and we had 3 of them that were warranting further analysis, and that's on chromosome 1, chromosome 3, and 12. And the one on chromosome 1, that's the LOC105374218, has been reported to have a response association with response to candacetin. Well, candacetin is a drug that is used to regulate blood pressure, and we know that blood pressure closely relates to kidney function over time. And then the GAL-NT6 gene in particular is a known tumor promoter for breast cancer and has also been associated with total cholesterol levels. Well, the relationship between breast cancer and kidney function is just an upcoming one, but we can still tell that because we're able to replicate one of the BCAS3 gene that is also related to breast cancer, so we think that there could be a probable association. And then the TRIM11 gene is a gene enhancer for the OBSCN gene that has been very much associated with creatinine measurement and also with serum creatine. So in replication, we were closely looking at finding out if some of the loci that have been determined with blood urea nitrogen can be replicated in our study. And yes, we were able to replicate 2 loci and 2 SNPs from the Europeans and the East Asian. Like I noted that these are the populations where blood urea nitrogen has been studied. And we had the HOXD11 gene, the TFCP2L1 gene, the BCAS3 gene, and the RNU148P and STC1 gene. Well, these genes have clearly, there are studies that have proven that they are related to kidney function and mostly in growth of the kidney. The BCAS3 gene is the one that is related to breast cancer and has been associated with almost all the biomarkers of kidney function. So our functional annotation results, we used MAGMA, we used FUMA, where we tried to determine functionality in about 53 tissues. And we had highest expression in the pituitary, kidney medulla, and the thyroid. Well, the pituitary produces the antidiuretic hormone that relates with kidney maintaining of water balance. The kidney medulla too regulates the amount of urea in the body, and the thyroid too has been reported to have relations with kidney function. Though our results were not statistically significant, but we could tell that there is biological relevance to kidney function using blood urea nitrogen as our biomarker. So this particular study, in conclusion, shows that blood urea nitrogen is a plausible biomarker for us to use on the continent to determine the underlying genetics, given that we're able to replicate what has been found in other populations, and also we get to see new loci that could be related to blood urea nitrogen. And then it can also help us inform any prevention and precise treatment strategies that can help us reduce disease burden, especially for chronic kidney disease. And also the fact that we're able to replicate what has been found elsewhere shows that BUN can really be used to help us determine what could be underlying for the genetics of continental Africans. So some of the future directions of this study is that, yes, we do need more continental Africans to participate, like I've shown. This particular study only considered southwestern rural Ugandans. Uganda alone has around 13 ethnic groups, so we only consider like a part of them. And now Africa has a lot more. So if we need to understand or come up with treatment strategies, we need participants from all over Africa. And then it's also a call for investment for Africans to invest in kidney genomics. yes, now that we know we can actually find out what is happening in kidney genomics, it's a call for us to invest money and understanding into kidney genomics, and also to encourage discussions on policy integration, because policymakers help us calling people to attend these studies and to participate in them. I'd love to acknowledge my supervisors, the Medical Research Council, members of the African Computational Group, the ISCB, for giving me an opportunity to present my work. Thank you very much for listening.
So does anybody have any questions?
Thank you so much for a fascinating talk. I'm very curious about, given the multifaceted nature of kidney function, with a study like this, are you able to collect any lifestyle factors, or what were your exclusion criteria for participants?
Okay, so for this particular study, some of what we considered was mainly age and sex. We did not consider a lot of maybe whether someone is a drunkard or a lifestyle. It wasn't really very much considered. Yes.
So nothing like medication?
No, it wasn't that considered. Yeah.
Thanks for the presentation. My name is Francis, and I have a question with regards to the statistically significant threshold that was decided on.
Yeah.
So my question is, what was the justification, and then the biological response to the type of interest?
Okay, so ideally, like I noted, that we did not get any SNPs at the conventional threshold. So we thought to ourselves that maybe there could be any informative SNPs. So we went ahead to actually do the false discovery. We thought of using the false discovery rate threshold, but we did not get any in this particular study. So we said we could just slightly increase the p-value and more like consider the top SNPs for the summary statistics that we got. Yeah, that was really, it was an abstract decision. Yeah. Yes.